Can you see the slide? Hello?
Yes, we can.
All right. So we are going to talk about parasitic weeds.
Yes.
You can see the slide.
Yes. Yes, we can see it too.
Okay. So the learning objectives are basically three. The first objective is that we want to be able to identify parasitic weeds and their symptoms on the host. The second one is to be able to understand the biology of the parasitic weeds. And then the third, to be able to understand the general control strategies. So this first weed is Striga asiatica. It is characterized by red flowers. So this is Striga asiatica with these red flowers. Striga asiatica on maize in the low veld. We have got maize which is being parasitized by this Striga asiatica. Again, this is Striga asiatica on sorghum. This is the plant with the red flowers. This is in the low veld. Again, sorghum infected with the Striga asiatica. This is the low veld area. Now, sometimes we can use polythene bags to grow striga when we are screening. In this case, we're screening maize varieties for tolerance to striga. This is Striga asiatica in Gokwe. You can see very short maize plants. Here, this field was infected with the striga. Again, this is in Gokwe. Probably you can see the farmer holding the striga plants. Again, this is in Gokwe, where you find that farmer holding the striga plants. Now, there are different strains of Striga asiatica in Zimbabwe. We have got the white flowered striga, which we find in the northern part of Zimbabwe around Mzarabani area. And the common one is red flowered. Another parasitic weed which we have in Zimbabwe is Striga forbesii with the pink flowers. This one is Striga Neroids. In Zimbabwe, it attacks tobacco, but it has been reported that it can attack cowpeas, especially in West Africa. This parasitic weed is known as Striga Monthica. This one is found in West Africa. It's the major problem. And also probably in East Africa, going beyond as far as Tanzania. You can see that it's quite a serious weed, especially in East and West Africa. You can see it is growing in the rows where sorghums are being grown. This one is Striga Neroids. In Zimbabwe, it parasitizes tobacco. Again, Striga Neroids. Here it is attached to the root, tobacco root. Again, we have got the roots of tobacco supporting Striga Neroids or Neroids. This is the field. I think this is in Mvuma, where tobacco was seriously affected by Striga Neroids, and it affects tobacco quality. This one is Electra vogeri. It attacks bambara nuts and the cowpeas. Again, this is Electra vogeri on bambara nuts or bambara groundnuts. Electra vogeri again on bambara nuts. This is Electra vogeri on cowpeas, which was planted in polythene bags. This was actually an experiment. You can see the leaves are damaged because of the presence of Electra vogeri. This one is a shoot parasite. It grows on the shoot. It's Cascuta campestris. It looks like a spaghetti. You can see it looks like spaghetti with the white flowers. It's Cascuta campestris. Again here, Cascuta campestris, which is a shoot parasite. This one is Ramphicappa fistulosa. It parasitizes rice. In Zimbabwe, we find it growing in the flare areas where rice is grown. Again, this is Ramphicappa fistulosa. Probably it's now shedding seeds. This one is the Mistru. It's a shoot parasite which grows on trees. Here it's a problem in the forests, but sometimes it plays a role in the ecosystem. Maybe in fruit trees it may become a problem. We are not sure. Again, the Mistral 2 attacking a tree. There's a paper written by Ndagurwa and his team about the role of mistral tours in Zimbabwe, where they did a study in semi-arid savanna southeast of Zimbabwe. You may read this paper if you want, if you are interested in these shoot parasites which affect trees. The broom rape or banji species, these are a problem, probably some parts of Ethiopia going into Saudi Arabia and around the Mediterranean Sea countries, Morocco, in North Africa, I think it is found, but we do not have reports of this parasite in this country. But we have to watch out, especially if we get seed from outside. We need to check it for this broom rape, especially in the plant quarantine departments. Now, parasitic weeds obtain carbon, mineral salts from the host via a special structure known as the ostoria. We've got local names for the witchweeds, which is the common name. Bise, Roombi, Isona. These are the local names in Zimbabwe. Now we can classify plant parasites according to where we find them attached to the host. If the plants are attached to the shoot, we call them shoot parasites, like the mistral toe and the Cascuta campestris. Alternatively, the parasites can get attached to the roots. In this case, we term them root parasites. For example, Striga asiatica, Electra vogeri, Striga forbesii, Striga Neroids. All of these are root parasites. We can also use chlorophyll to classify these parasites. One group, we call it the hemiparasites. Hemiparasites contain chlorophyll and are able to photosynthesize. For example, Striga asiatica. The second group is called the holoparasites. These do not contain chlorophyll at all, or they lack chlorophyll. These are the orobanchi. Orobanchi can attack tomatoes, horticultural crops, and as I have indicated earlier in the slides, these can occur in Ethiopia, Morocco, Saudi Arabia, and around the Mediterranean region. They are not yet a problem in Zimbabwe. Then we have Cascuta. Cascuta is the boundary between holo and the hemiparasitism. It has a bit of chlorophyll. Now, as far as crop yield losses are concerned, we should appreciate that 73 million hectares, probably more up to this stage, are devoted to cereal production in Africa. And this hectarage is affected by parasitic weeds, according to Mbubu in 1994. Sobon in 1991 estimated the yield loss, which ranged between 6 and 21%. Maize grain yield loss was up to 40%, according to Kim 1991. So cereals and grain legumes affected by parasites may actually fail to produce grain. So parasitic weeds could be a serious threat to food security in Africa. Here I've got a table of common parasitic weeds in Zimbabwe. We have got Striga asiatica, which parasitize maize, sorghum, millet, and sugarcane. Striga forbesii, again, it parasitizes maize, sorghum, millet, and sugarcane. Then Striga gesneroids, it attacks tobacco and cowpea. We are yet to receive reports of Striga gesneroids in cowpea. Otherwise, we always see it on tobacco. As far as Electra vogeri is concerned, it has been observed to attack bambara nuts and cowpeas. Whereas these Striga species have got red to pink flowers, Electra vogeri has got yellow flowers. Then we have Cascuta campestris. That one attacks lucerne. It's a shoot parasite. Then there's another one which is not appearing on this list, which is known as Ramphicapa fistulosa, which attacks rice. And I have it on this slide, where I'm going to indicate to you the plant families where we find parasitic weeds. The first plant family where we find the parasitic weeds is the Urubangasi. Under this family, Urubangasi, we have got Striga asiatica, Striga forbesii, Striga gesneroids, Electra vogeri, and Ramphicapa fistulosa. Then we have the plant family Cascutaceae, and sometimes known as the Convolvulaceae. This is where we find the Cascuta campestris. So most of the plant parasites in Zimbabwe fall under the Urubangasi family. Cascutaceae or Convolvulaceae, we have got one parasitic weed, the Cascuta campestris. Now, seed size is very important among parasites. Actually, the seed looks like dust. That's how small it is. But if you are interested to know the actual dimensions or the statistics about the weed seed sizes, number one, Electra vogeri has got 0.2 millimeters and it weighs 0.3 micrograms. This is very small. Striga asiatica, 0.33 millimeters length and 3.7 micrograms. Striga forbesii, 0.5 millimeters length and 12.4 micrograms. But the take home message is that these seeds look like dust. They are very small. As far as seed production is concerned, you can have one plant of Striga asiatica producing about 25 seeds per plant. Striga forbesii can produce 58,000 seeds per plant. These are high numbers of seeds from individual plants. No wonder why these weeds are difficult to control. In addition to high numbers of seed production, they can also last for a long time. They have a seed longevity which is about 20 years. These then are the seeds of Striga asiaticus under a microscope. This is what you see. They look like snails under a microscope. This is Cascuta campestris seed under a microscope. You can easily differentiate between Striga asiatica seeds and the Cascuta campestris seeds. Now, if you look at the world map, this is where we find these black dots. They are representing where we find the parasitic weeds. In North America, there's one place where we can find Striga asiatica. You can see that Africa dominates dominates in terms of parasitic weeds, as well as the Asian countries. This is actually with respect to Striga species. Of course, I said that around the Mediterranean, you have the Urubangi in Ethiopia as well as in Saudi Arabia. If we look at the agroecological zones of Zimbabwe, we find that we do find the parasitic weeds across all the agroecological zones. In fact, with this climate change, we are going to get probably more droughts. The parasitic weeds are likely to become a serious problem. So let us look at the life cycle of the parasitic weeds. Now, you start with the seeds, which look like dust. These seeds can not ordinarily germinate, just like other weed seeds. Of course, dormancy is one of the factors, but for a striga seed to germinate, you need to expose it to soil moisture for a period of 2 weeks, or you can say 1 to 3 weeks. And during this period, the temperature should range between 25 and 35 degrees Celsius. So you need moisture for maximum probably 3 weeks, but on average 2 weeks. And the temperature 25 to 35 degrees Celsius. It's like an incubation period. It's the preconditioning period. Once that has been achieved, you now need the presence of the host, which is going to be attacked. That host should produce germination stimulants. The germination stimulants, we call them strigalactones. as a group, but some of the examples are strigol, sogolion, sogolactone, and electrol, just to mention a few of these chemicals which can actually induce the germination of striga seeds. So once we get those germination stimulants coming into contact with seeds which have been preconditioned for 2 to 3 weeks, then germination will occur. The next stage after germination is that the the radicle of the parasite has to seek attachment to the roots of the host. And what is required for this process is that we should have what are known as hostoria initiation factors. These hostoria initiation factors are produced by the host. For example, we have got what we call 2,6-DMBQ, which was identified as one of the most common hostoria initiation factor that can actually cause attachment of the radicle to the host root. And a structure known as the hostoria is formed. The hostoria is like a conduit which will allow the passage of water, nutrients from the host to the parasite. And the parasite is going to utilize these for growth. Now, after attachment, the parasite can stay within the soil for 30 days. Then it will emerge. After emergence, it can actually take another 30 days to form flowers. At this stage, to form flowers, and then it will finally produce seeds, and that will complete the life cycle. So that is the summary of the life cycle of parasitic weed. Probably we may just have to go through again through these stages. We said we start with the seeds, which require a period of preconditioning. Preconditioning means exposure to soil moisture at a temperature ranging from 25 degrees Celsius to 35. And the period of exposure could be 1 to 2 weeks, but sometimes it can go up to 3 weeks. The chemical signals in the root exudates will be required after this preconditioning period. This is an example of the germination under laboratory conditions where we have seeds which were preconditioned for 2 weeks at the required temperature, and then they were exposed to the radicle of the host. This was under laboratory conditions. Then we got this kind of germination. You can see this is the attachment to the root. This is actually happening in petri dishes, in agar gel. And we have got hostoria formation on the roots. This is the striga attachment. This was actually, I took this from one of the students' project. Now, the germination stimulants, which we call strigalactones, we have got strigol, which was first isolated in cotton. Although cotton doesn't support plant parasites, it can produce strigol. Then we've got sogolion and the sogolacton. We can also have strigol analogs. These are actually artificial, like 2GR24. There was an attempt to make strigol analogs so that we use them like herbicides to cause germination stimulant, germination in the absence of the host, thus killing the parasite. This is electron, which is produced from the uh, roots. Ethylene can also be produced from the roots, but ethylene was also tried in America to try and kill the parasites in the absence of the host to cause germination, which is termed suicidal germination. Contact with the host root. We said the parasite forms hostoria as a contact point with the host. And for such a process to take place, we need a hostoria initiation factor. For example, 2,6-DMBQ or 2,6-dimethoxy-benzoquinone. Now, striga triggers the formation of this substance at the root surface. At the point of contact, you get this substance. Others, which are Hostoria initiation factors, you can have the role played by phenolics, flavonoids, cytokinins, and diazuron. All these may facilitate the formation of the Hostoria. This then is a diagram which is showing under a microscope the point of contact of the parasite and the host. Here's the diagram which is showing this Hostoria, which has got xylem. It's like a bridge. This is where what is used by the parasite to get water and nutrients from the host. So the period underground, the parasite is very small and lacks chlorophyll. At this stage, it is fully dependent on the host. Crop damage occurs at this stage. It may not be easy to control using mechanical methods. Anyway, the farmers cannot see it because it is underground. So the parasite emerge after 30 days. It takes 30 days after germination, 30 to 40 days after emergence, it goes into flowering, seed formation, then the life cycle is complete. So this is it on the life cycle. Now we want to have a look at the effects of the parasites on their hosts. Parasites are very small, but they can cause a big impact on the host. Number one, the effects could be variable because sometimes they are not detected, especially if the host is resistant or tolerant. But if it is susceptible, the host may die. Number two, infected cereals are characterized by very low stem weights and low grain yield. In fact, sometimes we use the stem weights as an indicator for the impact of the parasite. This is Striga asiatica. You can see these parasites along the crop rows. This is in Chundura communal area near Kwekwe. This is the symptoms of parasitic weeds. You can see wilting even if under wet conditions. So that could be a sign of the presence of the parasite. It can also reduce plant heights, like what happens to this SC513. On the left, the plants are very short compared to the right. If you check underneath the maize, you find these plants which are Striga asiatica. So one of the effects is standing of internode elongation. It can cause the plants to be very short, and it can also cause wilting symptoms even under wet conditions. There are similarities between the physiological effects caused by drought and those caused by striga, because usually you have abscisic acid accumulating in the leaves as a signal of drought or as a signal of the plant parasites. Striga reduce the rate of photosynthesis in the host. Striga tolerant cultivars are capable of maintaining photosynthetic rates, and striga can predispose sensitive crop cultivars to photoinhibition, light impairment of photosystem 2. So the presence of striga can cause damages to the leaves. So they won't be able to photosynthesize efficiently. Striga infected cereals also show lower stomatal conductance than uninfected plants. This simply means is that if you have got striga infection, the stomata tends to close. That's what happens. They tend to close the. Once they close, they need to limit carbon dioxide assimilation, and probably this is what leads to low photosynthetic rates. We've got instruments which we can use to measure photosynthesis, which are as follows: portable modulated chlorophyll fluorimeter, infrared gas analyzer, and perometer. I mean, you can use these instruments to detect the presence of this parasitic weed and what it does physiologically to its host. Number 7, we can, there is a hypothesis which says that there could be phytotoxic effects or phytotoxic substances could be supplied by the striga to its host. Now we want to look at how, because we are saying parasitic weeds take carbohydrates from their host. In fact, when they take carbohydrates from their host, they act like a sink. And we know that the fruit or seed or leaf could form a sink on a plant. So let us look at the source-sink interactions. This is in terms of carbohydrate movement. Electravogary host interactions. We can also look at Cascuta campestris host interactions. I mean, among these parasites, the movement of the carbohydrates can explain the impact of the parasite. For example, redirection of host resources to the parasite sink. Explain what is happening to the host. Parasites become strong sinks for carbon and nitrogen. So the host can have a reduced grain or reduced yield simply because of the movement of carbohydrates from the host to the parasite. This is quite obvious with Electra vogeri and Cascuta campestris. But in striga serial interactions, this is a bit complicated because striga plants are very, very small compared to the host. And if we say the carbohydrates have been taken by the parasite, and that accounts for reduction in growth, that may not explain what is happening. So parasite can make a significant contribution to its carbon requirements. In addition, the parasite can also photosynthesize, which makes even it difficult to say that the parasite has taken all the carbohydrates. Yet it can supply itself with through photosynthesis. But of course, the parasite biomass is a fraction of the observed difference between infected and uninfected plants. So the question is, where are all the carbohydrates going? In fact, carbon balance model indicated that only 20% of carbon produced was directed to the parasite, and 80% was unaccounted for, where you have a parasite in cereals. So we want to find out what could be happening to the 80%. Probably the low leaf area and the low rates of photosynthesis, which are a result of infection, could account for other carbon losses. Low stomatal conductance, because the presence of Striga asiatica in maize may cause the stomata of the maize leaves to close, thus limiting carbon dioxide entry into the leaf. This has got some consequences on the photosynthesis process, which produces the carbohydrates. Now, the other issue is that the presence of the parasites can increase photorespiration or photorespiratory metabolism that also contribute to the reduction in the rate of photosynthesis. We know that during photorespiration, the plant will switch to use oxygen instead of carbon dioxide. And the Calvin cycle is affected because the Calvin cycle normally uses carbon dioxide to produce carbohydrates. But instead, this consumption of oxygen will lead to the reduction of carbohydrates. And because we have got less carbohydrates, probably we will have less growth. Other things are like other changes are in the form of growth regulators. So we can have changes in host biomass allocation and the architecture. For example, infected plants have higher root shoot ratios and shorter internodes relative to uninfected controls. What we are saying here is that there is also the movement of carbohydrates to the roots, although it partially explains what is happening. For Electravogeri, this could be an important factor, or Cascuta campestris. Now, changes in abscisic acid, gibberellins, and cytokinins could cause architectural changes. Now, if you have architectural changes, it may mean that in maize, the leaves will be pegged together in such a way that there is going to be self shading. That's also reducing photosynthesis. And also ABA can cause the stomata to close and limit carbon dioxide, which is also essential for carbohydrates. And these have consequences on the growth of the plant, which becomes reduced. So these are some of the effects of the parasites on their hosts. So I think that is it uh, on the effects of the parasites. I will stop presenting here.